What's up guys, good evening my party. This is your NSEJS Biology Master Teacher Akshansh and welcome back to the next session for NSEJS 2025 Biology Basic to Advanced series. Now in the previous session we had learned a lot of things about the structure of a prokaryotic cell. Well, today we are going to wrap up the topic and I'll start teaching you about the eukaryotic cell. So without any further delay, let's start with the class. So in the previous class we had talked about the structure of a prokaryotic cell. We had in depth talked about all the major components of a prokaryotic cell. We had talked in depth about the cell envelope, which has further three parts: the outermost glycocalyx, the cell wall, the bacterial cell wall, which is made up of peptidoglycan. We had also talked about gram staining technique, gram positive and gram negative bacteria. Then the structure of the plasma membrane, we had talked about what is nucleoid, the function of the cytoplasm, ribosome, mesosome, chromatophores, etc., etc. Now the topics that were left were flagella, pili and fimbria. So let's start with the very first structure, that is the flagella. Okay, so let me just write the heading: flagella. So flagella, well, the singular term will be flagellum. So flagella or flagellum is a whiptail-like structure that assists in the locomotion of the cell. The bacterial flagella performs rotation type movement that brings about the backward pushing of the water that results in the forward movement of the bacteria. So I'll just write the same thing. Well, it is a whiptail-like structure. Whiptail-like structure that assists in cell movement. Correct. Now the next point is going to be that the bacterial flagella performs rotation type movement that brings about backward pushing of water that results in forward movement of bacteria. Yes, so very important point about the flagella. Now we can look at the structure of the flagella. Okay, so flagella, let's talk about the dimensions first, right? So flagella, I can write all the relevant points over here. Okay, so if I talk about the diameter, so it is about 0.02 micrometer. 0.02 micrometer. And if I have to tell you the length, so it is 1 to 7 micrometer. So the length is 1 to 7 micrometer. Yes. Now the bacterial flagella, it has three parts: the basal body, the hook and the filament. Okay, better I draw the structure for the same. So let's say this is the plasma lemma. This is what? So the blue line denotes the plasma membrane. Then I'll just draw thick line. This is the cell wall. Okay, so this is what? This is the cell wall. And with the help of, like, let's say purple color, I can draw the rest of the part. Okay, so let me just take another color. Okay, Okay, so we can do the labeling. So this is the filament. This is the filament. This part is the hook. The curved part, that is the hook, and beneath you have the basal body. This is the basal body. Okay, and one very important point that you have to remember, that the prokaryotic flagella, it is unistranded, equivalent to single microtubular fiber, and it is made up of the protein flagellin. In case of eukaryotes, the flagella is made up of protein tubulin, but in prokaryotes it is made up of the protein flagellin. Okay, so I'll write all these things. Let's say the flagella, it has three components. First we have the basal body, then we have hook and filament. Okay, so the flagella is unistranded, equivalent to single microtubular fiber, and in case of prokaryotes, the filament is made up of the protein flagellin. Okay, so this is made up of protein flagellin. Very important. Okay, so this is all about the structure of flagella. Okay, I can give you a quick recap. So flagella is a whiptail-like structure that assists in self movement. The bacterial flagella performs rotation type movement that brings about backward pushing of water that results in forward movement of the bacteria. Now, so we were looking at the structure. The diameter is somewhere around 0.2 micrometer, length 1 to 7 micrometer. Flagella, it has three components: basal body, hook, filament. The filament is made up of protein flagellin, and the flagella is unistranded, equivalent to single microtubular fiber. This is all about the flagella. Now we can talk about pili and fimbria. Okay, so let's continue. Better I'll make a table for you, that is going to help you a lot. So we are talking about pili and fimbria. Yes. So the first point is going to be the occurrence. The pili only occurs in gram-negative bacteria, but fimbria, it can occur in both gram-positive and gram-negative bacteria. Okay, so gram-positive bacteria, so for pili, gram-negative bacteria only. Right, so in NSEJS, a question can be framed like there was a bacteria that was having pili. Okay, so this statement will demonstrate that the given bacteria is a gram-negative bacteria. So that's how they can incorporate various concepts in a single question, and that can really make you more confused. Now the fimbria, gram positive or gram negative, it can occur in both gram positive or gram negative bacteria. Now we can talk about the number. So pili 1 to 4 per cell. Okay, pili 1 to 4 per cell. For fimbria, it is like they can be ranging between 300 to 400 per cell. Okay, so the fimbria 300 to 400 per cell. Okay, so now the most important thing, that is the function of pili and fimbria. So pili, it basically helps in conjugation, which is a type of sexual reproduction found in bacteria, whereas fimbria is meant for the addition. Fimbria is meant for the addition. Pili is a tubular-like structure. Fimbria, they are bristle-like structure. Okay, so I'll write helps in conjugation, and यहाँ पे it helps in adhesion. Okay, so pili, it is a tubular structure, whereas fimbria, so fimbria is bristle-like structure. Bristle-like solid structure. Okay, now pili, so if a cell wants to have pili, it need to have the F factor, fertility factor, that is in the form of the plasmid. Okay, so it requires presence of F plasmid. Okay, so the pili formation is controlled by F plasmid. I'll write the whole thing. Formation controlled by F plasmid. F stands for the fertility factor. Okay, so the cell should be F positive. It should have the fertility factor. But Fimbria, it is the formation is controlled by some gene in the nucleoid. Okay, so I can call it as the nucleoid gene. Formation controlled by nucleoid gene. Correct. So if you want to know about the dimensions, pili the length is 18 to 20 micrometer, but for Fimbria it is only 0.5 to 1.5 micrometer. That means pili are longer, Fimbria it is comparatively very shorter. Okay, so pili length is 18 to 20 micrometer, and Fimbria the length is quite less, only 0.5 to 1.5 micrometer. Okay, so here we are done with pili and Fimbria, and that wraps up the topic, the structure of the prokaryotic cell. Now we can talk about the eukaryotic cell. Okay, so the next topic is going to be about the eukaryotic cell. So the eukaryotic cell, what can be the best definition for the eukaryotic cells, right? So these are the cells having well organized nucleus and several membrane bound cell organelles. Okay, so eukaryotic cells, these are the cells with well organized nucleus. When I say well organized, that is the jacketed or the double membrane bound nucleus. Well organized nucleus and several membrane bound cell organelles. So except the kingdom Monera, we have other four kingdoms left, right? Protista, fungi, planta, and animalia. So except Monera, the members of all other kingdoms, they are eukaryotic. Okay, only the members of kingdom Monera, they are prokaryotes, rest are eukaryotes. Okay, so except Monera, all other kingdoms have eukaryotic organization. Now a term is there, that is protoplast. So what is protoplast? So well, the protoplast, it denotes the whole of the protoplasm of the cell. So what is protoplast? Protoplast, it denotes whole of the protoplasm in the cell. Denotes whole of the protoplasm in the cell, right? So the protoplast, it has four major components: the plasma membrane, the cytoplasm, nucleus, and vacuoles. Okay, so what makes up the protoplast? The protoplast, it has four major components. I'll write all of them. Okay, so let me just make this flowchart for you. First component is the plasma membrane. Okay, so in short I have written as like PM, plasma membrane. Then we have cytoplasm, nucleus, vacuoles. Okay, now cytoplasm. So cytoplasm has further two components: cytoplasmic matrix and the cell organelles. Okay, so cytoplasm, it can further be written as cytoplasmic matrix the ground substance, and cell organelles. So the cell organelles that are going to be there in the eukaryotic cell, right, they are going to be membrane bound. So the cell organelles, they can either be double membrane bound, they can be single membrane bound, or they can lack the membrane. So you must know the names of the double membrane bound cell organelles, single membrane bound cell organelles, and the organelles without a membrane, right? So this is going to be summary of everything. Well, in a nutshell, I'll write everything. So first of all, the cell organelles, the cell organelles, double membrane bound, then we have single membrane bound, organelle, without a membrane. I'll just shove this thing low. Yeah. Yes. So double membrane bound, mitochondria, plastid, nucleus, mitochondria, plastids, nucleus, single membrane, ER, Golgi body, glyoxysomes, lysosomes, vacuole, spherosomes, etc. So endoplasmic reticulum, Golgi body, lysosomes, spherosomes. One more, peroxisomes are also single membrane bound. So peroxisomes, glyoxysomes, vacuoles. Correct? So here you have the single membrane bound cell organelles. So organelles without a membrane covering, ribosomes, microtubules, microfilaments, centrosome or centrioles. Ribosomes, microtubules, microfilament, and centrosome. Centrosomes or simply the centrioles. Correct. So here I have mentioned the name of all the cell organelles that you have to read in your syllabus for your NSEJS exam, right? In NCERT, spherosomes, peroxisomes, glyoxysomes, they are not discussed in depth, but is at least we should have a basic idea about these cell organelles as well, because they are very important for your NSEJS exam. And also in previous years you will find 1 or 2 questions from these topics as well. Okay, so the next topic is going to be about the plant and animal cell, and all these cell organelles we are going to discuss in depth in the upcoming sessions. Okay, so here we have the structure of the plant cell and the animal cell that is given in NCERT class 11th NCERT. So you can look at these diagrams, they are very important. At least you should be able to memorize the diagrams. Okay, and next is going to be the difference between plant cell and animal cells on the various parameters. Okay, so first of all, cell wall. So plant cells, they have a definite cell wall that is made up of cellulose. In fungus, that is made up of chitin. In case of true bacteria, eubacteria, it is made up of peptidoglycan, protista. So some members have cell wall, some lack the cell wall. The again cell wall is going to be different chemically, and for the archaebacteria it is going to be made up of pseudomurine. Okay, so for plant cell I'll just write everything. Okay, so plant cell, they have definite cell wall, that is made up of cellulose, but the animal cell, there is the absence of the cell wall. So cell wall we are aware of. Next structure, that is the cell membrane. So cell membrane, for the animal cell it is the outermost covering, but for the plant cell it lies inner to the cell wall, right? So present internal to the cell wall for plant cell, but it forms the boundary of the cell for an animal cell. It is the outermost layer for the animal cell. So if a cell lacks the cell wall, it is going to be an animal cell, right? Then centrosome, that is the centrioles. So animal cells have centriole, plant cell lacks centriole. Very important. Okay, so absence of centrosome in plant cell, but presence of centrosome in animal cell. Very important. Okay. So done with now vacuoles. So vacuoles, the plant cell, they are going to have a very large central vacuole, but in animal cells the vacuole can be absent. If present, they are very tiny and numerous. Okay, so vacuoles, presence of one or more prominent vacuole in case of plant cell, but in case of animal cell, presence of small and temporary vacuole, right? Then the plastids, plant cells have plastids, animal cells lack plastid. Okay, so plastids are further three types: chloroplast, chromoplast, leucoplast. Okay, so the chloroplast, the kitchen of the cell. So plant cells, they have chloroplast. Animal cells, we are animals, and in animal cell we don't have chloroplast. Okay, so plastids, absence of plastids in animal cell, but presence of plastid in the plant cell. Then functional differences. So let's talk about the first difference, that is the size. So plant cells, they are usually bigger in size, and the animal cells comparatively they are smaller in size. Okay, so usually smaller with less distinct boundaries, usually larger with distinct outlines in case of plant cells. Arrangement of cytoplasm. So again, there is the vacuole in the plant cell, and the vacuole pushes the cytoplasm to the periphery. Okay, so the same thing is written over here, only a thin lining of cytoplasm which is mostly pushed to the periphery. Okay, because of the presence of the large central vacuole, the cytoplasm is pushed towards the periphery. In case of animal cells, so cytoplasm fill up almost entire cell. Shape, so it is polygonal for plant cell, but for animal cell, the animal cells are generally spherical. Okay, and finally the storage material for animal cells, it is glycogen. For plant cell, it is starch. So you have to memorize this table by heart. You are preparing for NSEJS, and I expect you must be knowing these differences, and you can expect a question from this part as well. Very important for you. Okay, so now we are going to starting with each and every single cell organelle. Let's start with the cell wall first. So if I have to define cell wall is outer, rigid, supportive, protective, semi-transparent covering of the plant cell. Okay, so it is outer, rigid, protective, supportive, and semi-transparent covering of plant cell. So this definition itself discuss about the function of cell wall, that it provides rigidity to the cell, it provides protection to the cell, it provides mechanical support to the cell, it is semi-transparent. Well, cell wall, it is completely permeable to water, it is completely water permeable. It also allows small size molecules to pass through it freely. Just if the molecule is very bigger in size, if it's a macromolecule, then only it is going to resist its movement. Okay, so this point you have to keep it in your mind. Next, about the composition. Next topic is about the composition of the cell wall. Composition. So the composition for the cell wall, the NCERT mentions for both higher plants and the lower plants such as alga. Okay, so for higher plants it is cellulose, hemicellulose, pectin, protein, and for alga it is cellulose, galactans, mannans, and calcium carbonate. Okay, so you have to remember this in this manner only. Higher plants, and we have for it algae. I'll just change the color of the pen. Yes, so cellulose plus hemicellulose plus pectin plus proteins. For alga, it is cellulose plus galactans plus mannans plus calcium carbonate. Now cell wall, it is made up to three parts. Okay, the cell wall, it is made up to of three parts. Okay, you will have the middle lamella, then you will have the primary cell wall, and then you can have the secondary cell wall. Okay, so we are going to write this thing that cell wall can have up to three parts. Can have up to three parts. Okay, the outermost one is the middle lamella. Then we have the primary cell wall, and then we have the secondary cell wall. So let's start with middle lamella now. So middle lamella, it is a thin amorphous cementing layer between the adjacent cells. Okay, so it basically cements down the cell, keep them together, hold them together. Okay, so it is thin amorphous cementing layer between adjacent cell. Okay, chemically it is made up of calcium and magnesium pectate. So made up of calcium and magnesium pectate. And then so the ripening of the fruits, one very important point, that during the ripening of the fruits, right, the fruits after they ripen, they become soft. So that softening is due to the partial solubilization of the middle lamella into a jelly like substance. Okay, so ripening of fruits, like the softening, I should say, the softening of ripened fruits, it is due to the solubilization, the partial solubilization of middle lamella. Partial solubilization of middle lamella. Now, these are all the important points about middle lamella. Next, we are going to be talking about the primary cell wall. So primary cell wall, let's talk about the structure of the primary cell wall first. So the primary cell wall, the structure looks like you have the microfibrils made up of cellulose, right? You have microfibrils made up of cellulose which are embedded in the ground substance or the matrix, right? So here what you have microfibrils made up of cellulose, these are embedded in ground substance or matrix. Ground substance, that is the matrix. Okay, so in primary cell wall you have the microfibrils made up of cellulose. So let's take a few steps back. Here we have talked about the composition for the higher primary cellulose plus hemicellulose pectin proteins. So where are these hemicellulose pectin proteins? So these substances make up the matrix. Okay, so this matrix, it is going to be made up of first water, the very major component, second pectin, hemicellulose and proteins, so especially glycoproteins. Now, and one very important point, that primary cell wall, it is capable of showing growth, right? It can show growth. So it can show growth. Right? That means only the young cells, right? The meristematic cells, the cells having the capacity to grow, right, to get elongated. So these cells, they only have the primary cell wall, so that as the size of the cell increases, more and more substances can be deposited to the cell wall, increasing its length, right? Increasing its size. Okay, so the primary cell wall, it can show growth. Secondary cell wall, it can never show growth. Okay, so secondary cell wall is always let down once the cell has attained its maximum size, right? When it doesn't require to grow, but it needs to get differentiated. Okay, so secondary cell wall formed in mature cell, right? So as the cell grows, as the cell tends to become more mature, the primary cell wall, it diminishes. Okay, and eventually it will diminish, and the secondary wall will take its place. So you have the primary cell wall, then you have the secondary cell wall, and then you have the plasma membrane. That means the secondary cell wall lies between the primary cell wall and the plasma membrane. This is what you have to keep in mind. So it is produced in mature cell, formed in mature cells, right? The chemical composition is very similar to that of the plant cell, but in addition to these substances, you have some other substances like lignin, subrin, cutin. Okay, so composition. Okay, I again write this thing: lies between primary cell wall and plasma membrane. Okay, chemical composition. The chemical composition similar to that of primary cell wall, but it also has some other chemicals, like also have other chemicals like lignin, subrin, cutin. So these are the most important one, etc. Okay, so here we have all the details about the plant cell wall. Okay, so I hope you have understood everything till here. We can do a quick recap. Okay, so cell wall, it is the outer rigid, protective, supportive and semi-transparent covering of the plant cell composition. So for higher plant cells, cellulose, hemicellulose, pectin and proteins. For algae, it is cellulose, carotene, manganese and calcium carbonate. Cell wall can have up to 3 parts: the outermost, the middle lamella, primary cell wall, secondary cell wall. Middle lamella, thin amorphous cementing layer between adjacent cells, right, made up of calcium and magnesium pectate. Softening of ripening fruits is due to the partial solubilization of the middle lamella. Then primary cell wall, so the structure, so microfibrils made up of cellulose embedded in the matrix or ground substance, that is made up of water, pectin, proteins, is the glycoproteins and hemicellulose, and it can show growth. Okay, so secondary cell wall, it is formed in mature cells, lies between primary cell wall and plasma membrane. Okay, chemical composition similar to that of primary cell wall, but it also has other chemicals like lignin, subrin, cutin, etc. Very important for you. Now the next component is going to be the plasmodesmata. What is plasmodesmata? So plasmodesmata, these are cytoplasmic bridges that allow the cells, the plant cells, to communicate with each other. It maintains a protoplasmic continuum. It maintains the protoplasmic continuums, and also the plasmodesmata, they have the tubular extensions of endoplasmic reticulum that are known as desmotubules. Okay, so these are all the essential points about the plasmodesmata. Next is plasmodesmata. Okay, so what are plasmodesmata? Basically, these are the cytoplasmic bridges. Cytoplasmic bridges between adjacent cell. Okay, so that maintains the protoplasmic continuum, that is the symplast. Maintains protoplasmic continuum, that is symplast. Okay, and it also encloses tubular extension of endoplasmic reticulum, which are known as desmotubules. Correct? We are done about the very first structure, that is the cell wall and plasmodesmata. Let's keep it till here only. From next class onwards, we will be starting with plasma membrane. Then we'll come to the endomembrane system. Then we'll talk about other cell organelles. So I think in upcoming 2 to 3 classes, this chapter will also get wrapped up. The unit of cell biology is very important for your upcoming NSEJS exam. Well, if you'll check the previous year's question paper, you will find that many questions have come from this topic, right? You can expect 2 to 3 questions maximum from the chapter Cell, the Unit of Life, Biomolecules, and the Cell Cycle. That's all from my side for today's class, guys. Thank you very much. We'll see you in the next session. Good night. Take care.